Before we do the actual simulation, I would just like to comment on the background of the model that we are using here. It's a model of the glycolytic pathway. It's of key importance to understand the dynamics of the glycolytic pathway because of its situation between the external environment provided by the blood flow and the internal environment in the cytosolic compartment, which produces things that via acetyl-CoA are being transported into the mitochondria for major ATP synthesis. And on the other hand, the ATP that is generated in the mitochondria and from glycolysis is being used in all sorts of processes. And the example shown here is membrane-bound ATPases. As such, as a metabolism that is central to provision of energy throughout whatever source of whatever energy you need, it means that the glycolytic flow must, of course, be highly regulated, and the way this regulation works is via self-organization of network dynamics. In addition to being self-regulated to a homeostatic point where the actual amount that is needed is maintained, it also needs to be adaptive. Then, depending on demand, more or less ATP needed or other sources of energy, then metabolic flux must be regulated up or down. And as such, that would be the maintenance of certain levels of the metabolites such that the ATP production occurs at a certain speed. And what we will see here is a famous example that dates back from the 1960s to research that detected that apart from maintaining a stable state, glycolysis also has the possibility to do variations around a stable state. And we are nowadays thinking of these variations in terms of information processing. So in terms of variations of levels that provide some information to processes connected to it further downstream. Here's an example that the way a pathway works can show surprisingly a switching between maintaining a state, and that's shown in the upper graph here, where there's a homeostatic state maintained within the cellular cytoplasm, but under slightly other circumstances, and this is experimental recordings, measures of the glycolytic state, in this case the absorbance that reflects levels of NADH, shows regular oscillatory processes, and this is a totally emergent phenomenon. And these emergent phenomena are something that we can capture only by simulation of the models and not infer from the network structure based on a priori principles. The final figure at the bottom shows that there's also different types of oscillatory states. So it's just the beginning of structure of temporal organization, which to this day we still don't understand very well, and which we're trying to understand by studying computational models in the first place. Okay, so much for an introduction. If we now come to the first part, which will be the use of the Simbiology toolbox, I think for this purpose I can take away my webcam. I have a bit more possibilities here. And I'll bring up my MATLAB environment. I've made the MATLAB command window very small because we're not going to use the command window at all, but by typing in, as you can see, I have done it previously, typing in the word Simbiology. I'll just take it again, highlight the word Simbiology, pressing enter or return. You get the graphical user interface that is the Simbiology toolbox. The first thing we're going to do, we're going to import the model that I just downloaded from the biomodels database and simulate it, simply to demonstrate this process of obtaining a model that somebody already developed and tested, and whether I can use it, whether they can share with me, such that I'm able to first reproduce their findings and then take it as a starting point to go forward and do my research on that model. So let's get the model into this program here. The functionality on the top left side, which says Add Model, allows us to pick from a menu with a couple of options like loading model from a PK library, that's pharmacokinetics library. And the reason it has a pharmacokinetics library might be worth mentioning at this point. The Simbiology toolbox has two kinds of specializations. The first thing is it uses systems biology type of models to describe systems level dynamics of biological processes, often with emergent phenomena, like we discussed oscillations in the first place, also switching phenomena, where one state switches into another, but also transient development as being seen during development or evolution. And therefore, they have systems biology models. In addition, there's a PK library that is specially dedicated to the pharmacological community, where they're using pharmacokinetic models, or pharmacokinetic combined with pharmacodynamics models, where you can combine mechanistic knowledge of what goes on in the organism once a drug is there with the information about how much of the drug is there in which place at a certain point in time. So this kind of pharmacokinetics is a specialty. If you want to look in it, you can find tutorials that help you go through it. So if we now proceed with choosing the option Load Model from File, then you can see what it finds on my desktop is two models that are there already, and the one called BioMD, Biomodels Database, with the number 42 is the one that we downloaded as the Nielsen glycolytic model. So if I click on it, and yes, it's an SBML file, we'll see that in a moment, and I open it, then it will load the model and show me a lot of things, a lot of information about that model in the first place. I just mentioned it that this has a special format, a markup language format called XML, but I refer to it as SBML. And this is because these models are stored in a specific type of markup language, which is the systems biology markup language. So just clicking on it will bring up a version of this content of the file, and it's just to show you what it looks like. So it's a typical markup language file that contains all the information related to this model in a way that it can be automatically accessed so that it's machine accessible. So it contains the author, the reference where it comes from, and if I, for example, search for an entity called NADH, yes, there's also information about NADH, and it tells you that this is a species, which in our context now translate to a node in the network, that it's being located in a special compartment, which in this case would be representing the cytosol, and it has certain specifics. It has a certain initial concentration of 0.6 millimolar in this case, and other information related to it, reference to the KEGG database, and so on. And if you just scroll further down, then the next variable or the next node of this network coming up, which is DPG and phosphoenol pyruvate and so on. All the variables used in the model are highlighted here, and similarly all the reactions are encoded here such that MATLAB understands it and can interpret it in order to generate the equations and to do simulations properly. So this is just for those who are curious how information is shared between different platforms. SBML markup language has become the standard of the community to share information about computational models of biological processes. So if we go back to the Simbiology environment, we can have a look. And what we can see here, that of the 3 tabs that are available on the top left corner of this user interface, when we select the model tab, on the very left-hand side, we have something that says Open. Open allows us to access the diagram. That is the network view of the model as it comes from the database. So if I pick, by clicking on it, pick diagram, then it opens a window and we get the network representation of this model in an interactive way, such that you can, if things are overloading, that you can sort of move them around and make them a little bit clearer, if that helps for imagining what's supposed to go on. So this has connections between the metabolites, which are here in gray, and if you click on them, they're highlighted in red, like pyruvate. And so the small circles with yellow background actually represent the reactions that represent the conversion of, in this case, ACA, acetyl-CoA, which is created from pyruvate. If we look at the downstream ways of pyruvate to acetyl-CoA, which is, of course, the one thing that will then be transferred to mitochondria. We can also, by clicking on one of the nodes in the network, we can also bring up the block property editor. The block property editor, which, if you select a certain node, shows you all the way with which it is graphically set up. So that's the properties of the graph here. And if you watched the biograph session 2 weeks ago, we explored how we can interactively deal with graphical features in biograph. Same thing you can do with Simbiology by looking at, say, if you want to change the shape with which that node is represented, you have a set of options, and you can choose a shape of the one that you like best. And similarly, you can change colors of things, like the background color. So if you want to change the background color and highlight one of them, you can click and select it to red, and it will then be highlighted. So that's up to you then to experiment with it and to be able to, in this network diagram view, to get the graph that you like best, the representation that you like best. So now we're not going to look so much into the graph properties, but what we want to do is we want to create an action window and actually use that model from the overview, from the table overview, use that model to first find out what it is that it represents. You can see in our table overview, we have all the reactions listed, and listed such in a way that they are fairly intuitive. So like it tells us in the reaction number 24 that AMP plus ATP together form 2 molecules of ADP in a certain compartment, and they follow a certain law, in this case something that you can later decipher and transfer back to its mathematical structure. So all the reactions are there. In addition, we get also the more important quantitative informations about, first, how much of each variable is there. Each of the variables has a certain amount that we start our simulation with, and that is highlighted. So if we pick AMP, is highlighted by a certain number. So it gives us, say, a millimolar concentration. It allows you to set in this number, and not having selected that tick box on the right-hand side allows the algorithm to make it variable, to change its concentration over time if a certain set of instructions tells you how to do that. And at the bottom are all the numbers that are supposed to be constant, all the reaction rate constants, and other useful numbers like the influx, if it is an open flow model, like in this case, where we assume glucose coming in from the outside and supplying fuel for the maintenance of a metabolic flux. Each of the parameters here has a number, and it's of course related to the reactions that have been defined in the other window that we saw previously, in the window up here. So you can relate one to the other without ambiguity. And specifically, that very last parameter at the bottom of the page called the flow is something that was controlled experimentally in the paper by Nielsen et al. 1998, from which this model is taken. And so having it under experimental control also means we want to explore what is the computational prediction if I change that number, what's going to happen, in order to find out whether that is consistent with experimental observations. So let's do a simulation. In our toolbar, we have the option to choose Add Task. And if I click on Add Task, it unfolds a window of options where the first of all is to simulate the model. And if you look at it, there's also more things that you can do. You can fit this model to a certain data set if you like. You can calculate sensitivities. I'll mention briefly towards the end of the session where that might be useful, and other things. And you can generate a report that's a log of the things that have happened during the simulation. So if I press Simulate Model, a new window will open up, the simulation window. And why do we need to simulate? Simulation means we put in all the assumptions, we put in our network structure, something that we learned how to do 2 weeks ago. In addition, we put in the kinetic information. These were the reactions, the description of the reactions that actually connect the metabolites. So these are all the enzyme catalyzed processes that convert one metabolite to the other. And we put in numbers that are associated with that reaction, telling us how fast these processes are. And with this quantitative information, a large amount of possibilities can happen. And we'll do this. We have set it up to show how this is going to work, but it's something that you cannot infer from just looking at the network alone. So what we need to do is we need to have MATLAB say, take the instructions, take the constants, and the set of variables that we have prepared for you, and now predict what if this was the situation in a moment, what would happen into the future. So simulate a few minutes into the future in order to find out what is the prediction of the model, what is going to happen in that simulation in order to predict what we might expect to see in an experiment. And of course, if you don't see it in the experiment, you might wonder whether something goes wrong in the simulation and whether either the constants are wrong or the reaction rates are wrong or the choice of nodes is wrong, and modify it accordingly. Right, then we do a simulation by, if you looked at the instructions here, taking the third option to unfold the menu that regulates the simulation time, and by clicking User Stop Time specific to this task, we can change that number and make a simulation time that is much larger than the one that is set up here by default. By way of a warning, there is a bug here. Actually, this simulation, the way the model is set up, the way the constants are interpreted, is not seconds, but it's actually minutes. So that shouldn't be saying seconds here. That's a bug that we actually reported, but it's not yet being addressed. If I say 1000, here's the stop time, so it means you simulate from start point 0 to 1000 minutes in that case. So you have to adjust that on the figure axis. This is supposed to represent experimental minutes. That was it. And if we want to generate a plot, we can unfold this figure here. And for this very simple setting that we are doing here for our first introduction, just a time plot will be sufficient. But you can see many more options are there. For example, the possibility to have a comparison of simulated versus experimentally measured data, to have sensitivities displayed as bar charts, and so on. So if we just stay with the simple time plot will be enough. And the good thing about the curated model is that we can now go to find the run button, which is somewhere in the center of the toolbar. And by clicking the run button, we set the simulation going, and we can see the output displayed graphically, the concentrations of each of the variables, which in network terminology is each of the nodes of the network, as a function of time. So first, before we go any further, we try an interpretation of what we see. We have on the horizontal axis, we have time. The vertical axis, we have the concentration of each component. And if we want to see which component it is, we can actually click on it, and then we'll see it's being highlighted here in that diagram over on the top right side. So that's fructose bisphosphate. If you have read the instruction, this light blue graph somewhere in the lower third of our graph, of our figure. Right, what would we see? What we can interpret here, that all of the concentrations initially go through a phase. If I just zoom a little bit in with my mouse cursor, I can select a part. Initially, there seem to be trends in the graphs, trends meaning that overall the concentration does not stay stable, but it shows fluctuations and increases its mean. Many variables increase their mean, others seem to stay more fairly stable. So that would be a time of transient evolution of the concentrations. If I double click on the window background, I will restore the original axis as done in the simulation. If I now zoom into the right-hand part of the graph, what we can see is that there's oscillatory output, but now at a fairly stable mean value. So while the concentrations go up and down in time and have a temporal pattern, the mean of this concentration fluctuation doesn't seem to change. So we could use the mean of the oscillation to characterize the new state that has been reached after this initial transient period is finished. And what we want to do for the simulation, or what you're then supposed to do with the assignment, if you want to work with this model. So the first thing to work with it after you have successfully done a simulation, you can actually change numbers. So you can guess if you have a flow of 0, that means you are simulating that no glucose comes into the model. So there's no supply of basically the feedstock of glucose from the outside, from the blood level. And if we go back to our simulation window and simulate it, you can see there's the prediction of steady levels of the compounds, most of them being 0, apart from 1 or 2 that are not equal to 0. And this is actually ATP and NAD. And the reason being that their synthesis doesn't depend on the pre-existence of a carbon sugar, but they are provided from another source. They're supposed to come from some other source, so NAD as a source for NADH, but no reaction has taken place because no glucose is there, so stays at a constant level, and similarly ATP stays at a constant level. So this is one of the experimentation. This is the extreme case when there is no no flow into the system at all. And what we can do is we can then, of course, try put in another number. Previously, I think we had 0.011, and if I change that to 0.05, means I do now a new simulation, assuming that the provision of glucose from the outside source has increased. If I leave all the model parameters the same, just clicking on this will do a new simulation. In that case of the selected window that I had before. And you can see that there's also an oscillatory pattern at this range, although the shape of the oscillation has changed, which we can see if I zoom in a little bit closer into that window. And this is now ready for you to explore what happens if you make things, for example, between those two extremes. That was a higher supply than before. Then we had previously the extreme of zero supply. And if we had 0.01 before, we can also look what happens between nothing and between too much and the oscillatory situation. And we can then try to explore all the possibilities of outcome. And I need to restore the original window to see what happens overall. We can see the transient is still there. There's still oscillatory output. But if I now zoom into the window, you can see the shape of the oscillation has changed dramatically. And we have here a representation of what is called so-called relaxation oscillations. Okay, that concludes this brief introduction. I leave it for you to further explore this interface and to check what else is there. You'll be able to then also do maybe more comprehensive. If you want to do multiple windows, you can use the trellis functionality. And I leave it for you to explore and find out how the graphical user interface can be used to provide the information that we